Good afternoon. My name is Professor Gene E. Ology, and I'm here to talk to you about the exciting field of gene therapy. You may have heard different things in the media about gene therapy and wonder what to believe. Is it a modern day miracle, capable of curing people of diseases who otherwise would have no hope? Or is it a slippery slope where scientists end up playing God? Over the next few minutes, I'm going to give you a brief overview about what gene therapy is, how it works, what it's used for, risks and ethical concerns, and why it's such an important field of study that needs to be funded. Although there are some risks, the benefits of gene therapy could be far reaching and could change the world of medicine as we know it. Let's get right to it. Let's start off by asking the most obvious question. What is gene therapy anyway? According to Mayo Clinic, gene editing, quote, involves altering the genes within your body's cells in an effort to treat or stop disease. Simply put, the process alters genetic material by inserting, replacing, or deleting a DNA sequence in order to treat or prevent various diseases or disorders. Types of gene therapy include gene addition and gene editing. Going further, there are two types of gene editing: gene silencing and gene correction. Gene addition involves adding a new functional gene into the body to help fight a disease or disorder. Gene correction involves replacing a disease-causing mutated gene with a healthy copy of the gene. Gene silencing is the interruption or suppression of the expression of a gene, which is essentially switching off a mutated gene that is functioning incorrectly. Each of these approaches introduces a different type of gene-based changes, but they all start the same way, by collecting cells from modification. Pretty cool, right? How about we take a look at how gene therapy is applied? Gene therapy can be carried out in one of two main ways: in vivo or ex vivo. In vivo gene therapy involves delivering a therapeutic gene directly into the body via a vector through an injection. What's a vector, you ask? Well, vectors are essentially vehicles designed to deliver therapeutic genetic material such as a working gene directly into a cell. Viral vectors, which are made from modified viruses, have proven to be superior delivery systems due to their precision, efficiency, and versatility. Ex vivo uses vectors too, but in this case, cells are removed from the body, modified with the therapeutic gene, and delivered back into the body via a vector. Interesting stuff. So we know the basics of what gene therapy is and how it works. What else do we need to know? Well, it's important to understand that there are two types of gene therapy: somatic and germline, with the former being more advanced than the latter. Somatic therapies target genes in specific types of cells, such as bone marrow or blood cells. Somatic cell modifications are non-inheritable, which means they only affect the treated individual. Germline modifications, on the other hand, are applied to embryos, sperm, or eggs, altering the genes in all of the patient's cells. Unlike somatic modifications, germline modifications would be passed on to future generations. There is a much longer history of research and much greater regulation for somatic therapies than there are for germline editing. In fact, the first somatic trials were published in 1991, while human germline editing studies have only began in 2014. Let me explain to you where gene therapy can be used by telling you the story of Ashanti da Silva. In 1988, 2-year-old Ashanti was diagnosed with severe combined immunodeficiency, also known as SCID, which was caused by a lack of the enzyme adenosine deaminase, also known as ADA, in her body. Without this ADA, her T cells started to die off, leaving her unable to fight off any infections. After many more traditional but unsuccessful treatments, she became part of the first gene therapy clinical trial in history, and a healthy ADA gene was delivered into her blood cells via a disabled virus vector in 1990. The treatment was ultimately successful, and Da Silva is now a healthy woman in her 30s. Since then, gene therapy has been used to treat disorders such as muscular dystrophy, cystic fibrosis, cancer, and certain infectious diseases. It is hopeful that complete cures can be developed for these and other disorders using gene therapy as well. Of course, along with the potential benefits of gene therapy, there are some risks as well. For example, since the vector used to carry the new gene is an altered virus, it is possible that the immune system may attack it, which could potentially cause serious damage. It is possible that the virus may also regain its original ability to cause disease, which may result in infection. The virus may also target the wrong cells, possibly causing cancer, or the new genes may enter the wrong place in the DNA, and a tumor may form. Hopefully, as time goes on and research becomes more efficient, these risks will lessen. The more we learn, the more we can minimize these potential risks. Earlier, I mentioned that you may have heard some controversy in the media about gene therapy, so let's talk about that a little bit. Now, somatic gene therapy has been around for decades and is heavily regulated, so it has quite a positive reputation with the public. Germline gene editing, however, is much newer and much more controversial. Since genetic changes created by this type of gene editing would be passed on to future generations, it has raised ethical concerns for some people. Because people who would be affected by germline gene therapy have not been born yet, they can't choose whether to have the treatment. This raises many questions, such as who decides what uses of gene therapy are good and which are bad? Who decides which traits are normal and which are genetic disorders or disabilities? Due to the high cost of gene therapy, will it only be available to the extremely wealthy? Will society look down on those who are different if the use of gene therapy is widespread? Will this cause the advance of techno-eugenics, where the technology is used to improve rather than fix in an attempt to create genetically superior children? These are all important and legitimate questions and need to be considered with great care. It is imperative to proceed with caution to ensure that whatever steps we take towards advancing gene therapy are taken for the benefit of humankind rather than its exploitation. Now that we've covered the basics of gene therapy, let's have a look at someone who's working in the field. Gene therapy is a field that is growing exponentially with many exciting career opportunities in areas such as research, testing, and project management. One such example is wife and husband team Dr. Jean Bennett and Dr. Albert McGuire. Bennett graduated with honors from Yale University in 1976 with a Bachelor of Science in Biology. She followed this with a Doctor of Philosophy in Zoology, Cell and Developmental Biology from the University of California, Berkeley in 1980, where she first began developing molecular techniques for gene editing. She left this position in 1982 for Harvard, where she studied human genetics, finally obtaining her doctorate in medicine from Harvard in 1986. At this time, she was awarded a career development grant from the Foundation Fighting Blindness to begin research on gene therapy for retinitis pigmentosa, or genetic blindness. Dr. McGuire graduated from Harvard University with his MD in 1986, then finished a surgical internship at Yale in 1987, followed by his ophthalmology residency in 1990. At the time doctors Bennett and McGuire began research on gene therapy, there were no identified genes known to cause vision loss or blindness. However, researchers identified one sight impairment gene, RPE65, in 1993. Bennett and McGuire then utilized gene therapy to target this gene in 3 dogs who all suffered from severe vision loss. The therapy was successful, restoring the vision in all 3. Bennett and McGuire actually kept 2 of the puppies. The trials on the dogs led to successful treatment in people beginning in 2007. They named their therapy Luxturna, which became the first approved gene therapy for inherited disease in the United States and Europe. Hopefully, I have been able to provide you with some understanding of the mechanisms and importance of gene therapy. At present, the field is at something of a tipping point. Decades of research have laid the foundation for exciting new technologies. This has led to an exponential rise in new gene therapies. In fact, there are more than 2,000 gene therapies in development worldwide targeting dozens of areas, such as cancer, neurological, blood, immunological, cardiovascular, and infectious diseases. However, there is still some hesitancy for funding gene therapy. The great expense makes governments of first world countries hesitant to fund it, and governments of third world countries unable to do so. The ethical concerns around playing God also give some people pause. However, the enormous benefits that can arise from the use of gene therapy certainly outweigh the drawbacks. Gene therapy has the potential to give long-term relief to certain medical conditions that otherwise would be more difficult to treat and could possibly be an option for conditions which do not respond well to other more traditional treatments. It can also give hope to those for whom all hope was lost. If anyone understands the importance of funding, it would be therapy pioneer Dr. Jean Bennett, who, when asked how it felt to cure a child of blindness, replied, quote, "It's absolutely indescribable. Each time I've seen this happen, it absolutely makes me want to cry." Proper funding of gene therapy will allow more of these indescribable moments. I hope you enjoyed this video. and we're able to gain a greater understanding about the importance of gene therapy. For more information, follow one of the provided links. Thank you so much for watching.
Deoxyribonucleic acid contains your genetic code, DNA, a molecule that makes up all your chromosomes. Double helix structure, a two-stranded spiral. Set of instructions for your cells so that they know what to do, like sender signals and magnets.